Hi, welcome to meiosis and the issues around meiosis. This will be our last lecture online for cell division. Hopefully this has helped, and we're going to now look at some of the other issues that are associated with meiosis. Hi there. And so first, asexual reproduction. Asexual reproduction is a function of mitosis, and it's where you duplicate the exact set of chromosomes. We see this in roots and shoots of plants. We also know that offsprings who are identical using mitosis as a form of reproduction are called clones because they're exact copies. It's very rapid and very effective. Unfortunately, it leads to less diversity in the population. Let me share with you some examples. Many single-celled organisms will use budding or splitting or this idea of meiosis to replicate. In the examples you see here, this is an example of what we call budding. This is a hydra, it's a freshwater animal, and the offspring is growing off. It's a clone of the cells next to it and becoming an adult. In this case, these are aphids. These are small organisms that suck the juice out of plants, and they are an example of parthenogenesis. In this case, the female here has given birth to females who are sterile, and it turns out that she did not require the male whatsoever. So this is a natural function in which the females produce offspring who are genetically identical to the parent. But unfortunately, because of that process, they're sterile. Okay, let's look at sexual reproduction. Sexual reproduction is the formation of a new individual from two haploid cells or gametes. Fertilization takes place between these two nest cells and forms what we call a zygote. The gametes for fertilization usually come from the eggs in females and sperm from males. Both gametes are haploid and only have a single set of chromosomes, and they are random in terms of if those chromosomes are from the mother or the father. When they do form in this zygote, they make a diploid chromosome, returning them back to their normal chromosome number. And meiosis is a process to do this conversion of diploid to haploid in the areas where gametes are produced. So this is just a picture of sperm and one egg, so you get a nice idea about the size difference. But it's costly. It requires a large amount of energy, requires intimate contact, therefore it provides a route for infection via parasites or bacteria or viruses. It's very costly because you're only passing half of those genes on to your offspring. And males have the expensive luxury. In most species, they contribute little to the rearing of the offspring. Therefore, why have males? But the good part is that it increases genetic diversity by shuffling the genes in meiosis, by rearranging chromatids in something we call crossing over, and the fertilization then combines two separate individuals' genes. So by doing so, you increase the genetic variability in these species. DNA backup and repair is another example. Because of asexual reproduction, any mistake that is made, that mistake is then passed on in the next set of generations. However, with sexual reproduction, if a mutation occurs in one chromosome, then the other chromosome would have a backup if it's not damaged. And sexual mechanisms, especially recombination, are used to repair those damaged DNAs. So we have a built-in proofreading system. Okay, so we were talking in class about the life cycle of the organisms, and there are two sets of cells we talked about, one called somatic cells or cells of the body, and another one called gametes. These are the cells that are used for reproduction. And in doing so, it raises an interesting question about how mistakes or mutations can cause problems in the future. So we're going to use somatic as cells other than gametes, and gametes are the place where eggs and sperm are produced. And so we're talking about these chromosomes and how you can then see chromosomes. And that process of taking a picture of chromosomes for medical purposes is called a karyotype. And what we can do is stop the cells during metaphase, take a picture of them, stain them, and then take this picture and then organize them based on their shape, length, and banding pattern. Here's an overview of what we do is you just take some cells, you then pop the cells using a hypotonic solution, you then stain the cells, and then you place them under a microscope and take a picture. And then you begin to see the actual chromosomes of a human, and these are homologous chromosomes because they're chromosome 1. They have the same banding pattern and the same color stains. And here's a real picture of a human female chromosomes. You'll notice there are 43 pairs of chromosomes. 22 of them are somatic or cells or chromosomes because they are for the body, and they're sex chromosomes, which are right here, which are X or Y. In this case, since it's a female, it has two X chromosomes. Here's the example of a male. You'll notice that the male chromosome has one X and one Y, and that one Y is a very small chromosome compared to most of the others. However, it's important for making males males. So the question is, what happens if there's a mistake in one of these chromosomes? And these are called meiotic errors. So some of them we're going to see is called non-disjunction. That's where homolog chromosomes will not separate. Normally they're lethal, but there are some examples where they're not lethal and cause different problems. If you get an extra chromosome at number 21, we call that trisomy 21 or Down syndrome. If those mistakes or non-disjunctions occur at the sex chromosomes, then they're either called Turner syndrome, where they have only one X and no Y or another X, or Klinefelter's, which is two X's and a Y. A third kind of chromosomal damage is called translocation or deletion. This is where a piece of chromosome is literally picked up and moved to another part of the chromosome. This is an example of trisomy. You'll notice that here at chromosome 21, I have an extra set of chromosomes. So these two must have moved together. These two must have moved together because they're identical. And then this one came from the other parent. So you end up with 3 chromosomes instead of the 2. And this would be diagnosed as Down syndrome. And that should conclude everything here.